Okay, and we're live. Hi, everyone. Welcome. Thanks so much for joining us. Welcome to another MSU Science Festival afternoon science snack. I'm Catherine with the MSU Science Festival. I'm joined by Roxanne Troon from the festival as well. And today we're also joined by Jason Holohan. Hi, Jason. Welcome.
Hi, thank you for having me.
We're glad to have you. So before we dive in here, do you mind introducing yourself, telling us a little bit about what you study and what we'll be talking about today?
So my name is Jason Holohan. I'm a medical laboratory scientist. I work at the Michigan Department of Health and Human Services bacteriology laboratory. And today we're going to talk about antimicrobial resistant superbugs.
Great. Okay. Well, let's go ahead and dive right in and get started.
Okay. So yeah, like we said, we're going to talk about superbugs from the public health lab. And we've all heard of these evildoers, right? Periodically, they grab sensational news headlines like wreaking havoc in hospitals, destroying patients' lives. Their invisible, unseen nature makes them a disturbing and unsettling enemy. So this afternoon, we'll make the invisible visible and hunt down these superbugs. Just to reintroduce myself for anyone who's recently joined, my name is Jason Holohan. I'm a public health microbiologist at the Michigan Department of Health and Human Services. I work in the reference bacteriology laboratory. So first of all, when we talk about superbugs, we're not talking about cockroaches and capes. I know that would be really cool, but it's hard to find capes that small. When the media or scientists talk about superbugs, we most often are referring to bacteria and not insects. So what are bacteria? Well, bacteria are everywhere. There's an unseen world around you. Billions and billions of bacteria surround you and even live inside of you. There are more bacterial cells in and on you than there are your own cells in your body. They actually outnumber you about 3-1. You are essentially a walking bacterial metropolis. The bacteria you live with come with many different names. Most bacteria we encounter in our daily lives are called non-pathogenic or commensal. And our bodies are covered in this type of bacteria. Some of them are actually very helpful for our health. You can think of these bacteria as everyday citizens just living their lives on the crowded metropolis that is your body. Another kind of bacteria, and we can think of these as the common criminals in this plot, are called pathogens. These bacteria are nasty and can cause serious diseases, but with a trip to the doctor, some laboratory tests, and antibiotics, they can often be treated. So, antibiotics. We treat bacterial infections with antibiotics. Antibiotics are a wonder drug that are responsible for extending the average human lifespan by about 10 years. Antibiotics and our past victories over bacteria are one of the pillars of modern health and longevity. Antibiotics are special because they recognize and target only bacteria, leaving human cells relatively undamaged. Now, that's not to say these antibiotics are completely safe to use. Some have certain toxicities to humans, especially in our kidneys and livers. But when you think of the destruction that they bring to bacteria versus the relative safety to you, it's truly a miracle. There are lots of different antibiotics available, and different antibiotics work better against different bacteria. So determining what bacteria is causing an infection and then the antibiotics that will fight it are an important task for the microbiology lab. So today we're going to talk about bacterial superbugs. But what makes a normal pathogen super? Well, a superbug is a common pathogen that has acquired special antibiotic drug resistance. And you can think of these bacteria as a normal criminal that has gained a superpower. These bacteria are the evil supervillains of this story. And frankly, with everything that's happened here in the last 3 months or so, we may need to make a new supervillain category for this super virus, the SARS-CoV-2 coronavirus that causes the disease of COVID-19. And that brings up another definition that I want to make clear, the difference between bacteria and viruses. So bacteria are alive. For the most part, they can replicate by themselves and they can live their own lives. But some of them do turn to the dark side and make us sick. Viruses, on the other hand, are neither alive nor fully dead. They have no ability to replicate on their own, and they must hijack living cells to replicate. Viruses are everywhere that living cells are. There are even viruses that infect bacteria, and these are called bacteriophages. And because they aren't really alive, viruses can be even harder to treat than a superbug bacterial infection. At least with most bacteria, we have antibiotics and drugs that will stop or kill them outright. But since viruses use our own cells against us, they can be very difficult to treat. And that's why the current treatment for COVID or the current solution to stop the SARS-CoV-2 virus is to try and starve them out by limiting its food source, which is us, by socially distancing and isolating ourselves. But let's get back to bacteria and how does the laboratory find bacteria? So if you're sick, a doctor will collect a sample from the site of the infection, like a swab of a skin infection. They'll collect urine or blood and send it to a microbiology laboratory. Once the sample gets to the lab, the microbiologist will put it on bacterial growth media called agarose. Different bacteria prefer different agarose, so every lab stocks all their favorites. Bacterial growth agar is a bit like a stiff or a firm jello in consistency. So then the lab lets the bacteria grow up overnight in an incubator set at human body temperature. And the next morning we wake them up and see what's growing. And after initial testing of the bacteria, sometimes additional work needs to be done. And if that's the case, that's where the public health lab where I work may become involved. So how does the public health lab fit in? Local hospitals and laboratories act as the front line of the public health system. They do the initial testing of microbiology samples, and if their testing detects a pathogen of special interest, the state public health lab may become involved. So I often describe the work done in my laboratory as things that hospital labs either can't do, won't do, or is just outside of their focus. Meaning the public health lab does testing that is often higher complexity, or sometimes we work with more dangerous pathogens. We also do a lot of testing that is important on a statewide or population level, but might not be important to a specific individual's care. And of course, the main federal lab, the CDC, sits on top of this pyramid. They coordinate resources and information on a national level. So the state public health lab often acts as a middleman between the CDC and local institutions, sharing specimens and information as needed. So to make this point on sharing isolates and knowledge in the public health system more relatable, I'll use the example of a foodborne outbreak. So bacterial foodborne illness is a large part of the testing done in my laboratory. And whenever you hear about a food that's been connected to an outbreak, that data comes from a public health lab. So salad and lettuce often hits the news as a source of bacterial outbreaks. So let's say you eat a contaminated product and you become sick. You'll probably go to your doctor and your doctor will tell you to collect a stool sample and that gets sent for testing. The initial testing would be done at a local facility, a local hospital. And once a pathogen of interest is isolated, it's passed to the public health lab for further testing. And if results of that testing reveal something important, like other people getting sick from that same strain of bacteria or the same food source, we would share that data with the CDC and other local infection prevention folks. Of course, the news media may also get involved, and that food would be recalled, and hopefully we can stop the spread of the infection and end the outbreak. So the CDC has set up a similar system for the detection of antibiotic superbugs in hospitals called the Anti-Microbial Resistance Network, or the ARLN. Superbugs can be a big problem in clinical settings. Very sick long-term care patients are the most susceptible to superbugs, and they often get transferred between different institutions that can then spread the infection. So once a superbug is suspected at an institution, it would be sent to my laboratory, and further testing and confirmation can be done. Alerts can then be sent around to all the relevant institutions that a superbug is circulating, and prevention measures can be put into place, preventing its further spread. This network also works on a national scale. The ARLN network is operating in all 50 states, and there are 7 regional labs doing enhanced testing. And as you can see, Michigan is the National Tuberculosis Molecular Center for the entire country. Tuberculosis is still very much a problem, and in particular, preventing the spread of multi-drug resistant superbug tuberculosis is very important. And Michigan is at the forefront of this testing and prevention. So here is your state public health lab. This is where I work hunting down bacterial superbugs. Lots of other great work is being done in this building as well. We have a large virology section working with all the viral pathogens of interest. Of course, this is where some of the first COVID testing for the state was done, and we are still working hard and doing a lot of testing in this building. Also, because life doesn't stop, this is where we screen newborns for inherited diseases immediately after birth. So if you've had a recent pregnancy, or if you remember your baby getting a heel stick in the hospital, those cards, those blood specimens are sent here for testing. So let's go inside for a minute. Here are 2 of our super scientists at work. They are growing and working with the bacteria to study their super secrets. And here on the left, you can see our incubators for growing bacteria overnight. And on the right, you can see some stacks of agarose plates used in the culture process. In these pictures, a technologist is preparing cultures in what's called a biological safety cabinet. This is a piece of safety equipment that circulates air in a special way that keeps any bacteria or contamination away from the scientists. And so biological safety is very important at the state lab. And depending on the type of cultures we have that we work with, we have a variety of personal protective equipment to wear. So let's hunt down some of these supervillains. The 3 supervillains I spend the most time with and that we're going to talk about today are Staph aureus, carbapenemase-resistant Enterobacteriaceae, and gonorrhea. So let's get started. So the first bug we'll hunt down is Staph aureus. Staphylococcus aureus can cause a huge range of diseases, from relatively minor skin infections like pimples or small boils to life-threatening diseases. So now, not every staph roaming the streets is a super staph. It takes a certain set of skills for staph to become a superbug. So this is what Staph aureus looks like growing on some agarose in the lab. But how do we determine if it's a common criminal or a supervillain? So first, the lab tests the staph for antibiotic sensitivities. To do that, we cover these agar plates with the bacteria. Next, we put little cotton pads containing different antibiotics on the plate. We then incubate the plates overnight, and throughout the night, the antibiotic will seep into the agar and react with the bacteria. In the morning, we'll take the plates out and see what's growing. So as you can see, the isolate on the left has large zone sizes, And this would probably be interpreted as susceptible, meaning the antibiotics here worked and they stopped the bacterial growth. This would be like your common criminal. This bug may make you very sick, but treatment should be relatively easy. The isolate here on the right has very small zone sizes. You can see the antibiotics are not fighting the bacteria very well. This isolate is drug resistant and may be classified as a superbug. This bug is definitely going to be much more difficult to treat. So the antibiotic resistance patterns that we see in bacteria are all kind of held and controlled by the bacteria's DNA. DNA being the instruction manual for life. So all the information that makes you, and Staph aureus can be found in DNA. So nowadays, many labs use advanced methods such as PCR to study small bits of the bacterial DNA to study its super secrets. So PCR is a process in which the lab, we look for just small bits of DNA that would correlate to resistance or whatever we need to look for. So to determine the super status of Staph aureus, we then look for small bits of DNA that would indicate super status and resistance to antibiotics. So the future of this testing, which the CDC and state public health labs are all actively working toward, we would be able to read the entire bacterial genome through a process called whole genome sequencing. Basically, we use a much larger magnifying glass to find all of the bacteria's super secrets. So next, we'll discuss gram-negative bacilli villains. Gram-negatives are a very large group of bacteria that span the whole range of bacterial citizens, from commensals to pathogens. The criminals in this group can cause a huge range of diseases from food poisoning, wound infections, urinary tract infections, and blood infections. And the problem with this group is their habit of swapping and sharing superbug DNA that would make them resistant to the antibiotics. And in fact, antibiotic-resistant bacteria are not inherently more virulent. The problem with super gram-negatives is the challenging nature of their diagnosis and treatment. Appropriate treatment is often delayed while the superbug's super secrets are uncovered by the lab. So when microbiologists talk about bacteria, we often discuss what we see through the microscope. So a very common stain that we use to better visualize bacteria is called the gram stain. And that divides the bacterial world nicely into gram-positive, or purple, and gram-negative, or pink. So this distinction often also influences what kind of antibiotics will work against them. We also talk about the shape of the bacteria. If they look like little rods, we call them bacilli. And if they look like little circles, we call them cocci. And these are two of the most common shapes, but there are many more. Bacteria come in all shapes and sizes. The last bacteria we talked about, Staph aureus, is a gram-positive cocci. So it's a little purple circle. The next family that we're going to talk about are the gram-negative bacilli, so they're little pink rods. And here are two gram-negative bacilli growing on a colorful agar that we use in the lab called McConkey. So the bacteria on the left, E. coli, is pretty emblematic of the gram-negatives. E. coli comes in all different flavors: good, evil, superbug. It all depends on the genes or the DNA that it has picked up. E. coli without super DNA lives happily as a productive member of society in your gut. Everybody has E. coli in them. However, even this benign E. coli can cause infections if it gets into the wrong part of your body. So some E. coli has picked up criminal genes like E. coli 0157H7, which has a nasty toxin gene. E. coli 0157H7 often comes up as a foodborne diarrheal pathogen, often in undercooked ground beef, or sometimes it gets into salad and lettuce. But the worst is when E. coli picks up superbug carbapenemase-resistant DNA and becomes a supervillain resistant to most antibiotics. So these supervillain superbugs have been called nightmare bacteria by the CDC. These gram-negative bacteria are resistant to a group of antibiotics called carbapenems, which carbapenems are kind of the last line antibiotic to fight these guys. And they produce an enzyme that actually breaks up that antibiotic, rendering it completely useless. And it wasn't too long ago that they weren't even identified in this country. So back in 2001, only one state had reported a confirmed case of a carbapenemase-resistant superbug. But by 2013, they had spread across the entire country. So to test for the presence of these resistance genes, we use the same techniques as we did with Staph aureus. We test for antibiotic susceptibility grown on agar, and we use PCR to find the super DNA in the bacteria. But the reason that this antibiotic resistance can spread so fast is that these bacteria can share their super DNA. The super DNA is stored on what's called a plasmid. And these plasmids are small bits of DNA stored outside of the main bacterial genome. And so bacteria tend to be a little loosey-goosey with their genetic elements, and they can pass DNA to others. And these plasmids can be easily shared between bacteria, which spreads the super DNA faster. So this is almost like Thanos handing out infinity gauntlets like Oprah hands out cars. You get a gauntlet, you get a gauntlet, everybody gets a gauntlet. So the last supervillain that we're going to talk about may be the only one strong enough to take down James Bond. That's right, drug-resistant gonorrhea. So Neisseria gonorrhea, or GC, is a common bacterial STD. GC is never normal flora. It's never an everyday citizen in your body. It's always at least a common criminal. This organism tends to infect it can infect all of the mucous membranes in your body. And if it's left untreated, it tends to migrate to your joints and causes nasty joint infections. So here's GC growing in the lab on its favorite agar called chocolate agar. And it looks really wimpy. And in fact, that's part of the problem for labs that work with Neisseria. It doesn't grow that well in the lab. So often to identify gonorrhea in clinical samples, labs often go straight to looking for DNA with PCR. So here's a timeline from the CDC about the increasing drug resistance of GC. As you can see, gonorrhea keeps growing stronger and collecting drug-resistant mechanisms. Gonorrhea used to be very easy to treat, as you can see with this World War II era picture. We used to be able to cure GC in 4 hours with just penicillin. But penicillin doesn't work anymore, and here we are today. We only have one drug cocktail left to treat gonorrhea with, and we are quickly approaching untreatable super gonorrhea. So my question is, what's next for poor James Bond? So late 2019, the CDC released this publication of antibiotic-resistant threats. And for anyone interested in further reading, I would highly recommend this pamphlet. It goes much further in depth than I did tonight, but it goes into depth in a very accessible way. And here are some of the bugs that I work with in my lab and that we talked about. And as you can see, we only covered a fraction of the organisms on this list because I only work with a couple of these bugs. This problem is simply too big for one man to solve. And how big is this problem really? On the money side, the CDC estimates as high as $20 billion in excess direct healthcare costs, with additional costs to society for lost productivity as high as $35 billion a year. That's right, those numbers were B as in billions. And of course, more importantly, there are human lives behind those dollar signs. The CDC estimates that in the United States, more than 2.5 million people are sickened every year with antibiotic-resistant infections, with at least 35,000 dying as a result. On a global level, the numbers are much worse. And currently, resistant bacteria kill about 700,000 people every year, which is less than but similar to worldwide deaths due to traffic accidents or diabetes. And some future estimates put the future death toll at 10 million people a year by 2050. And that number may sound large, but it is very plausible if we truly enter a post-antibiotic era. So 60 to 80 years ago, before the wide-scale discovery and adoption of antibiotics and vaccines, bacteria and viruses were the #1 killer of Americans. Things like heart disease and cancer only became major killers after people started surviving infectious diseases and living long enough for those to become a problem. So here we are really on the threshold of entering this post-antibiotic era. And in fact, it's very nearly here. You and I are living in a time when some of these miracle drugs are no longer performing their miracles and lives are being lost to this microscopic enemy. So the time for action is now, and we can all be part of the solution. And so what can be done? The problem of antibiotic resistance has been decades in the making, and the solution is not a quick nor an easy one. The public health system where I work, we are all working hard at prevention, identification, sharing data, but more can be done and more is actively being done to try and do better. Patients and the public can help by following antibiotic prescriptions and finishing their course of prescribed antibiotics. And if you have a cold or a flu virus, please don't ask your clinician for antibiotics. Antibiotics are not effective against viruses, only bacteria. Taking antibiotics unnecessarily help breed these antibiotic-resistant superbugs. There are other things that can help. Common sense hygiene measures while you're sick are always helpful. Proper hand washing can go a long way in stopping the spread of infectious material. And frankly, I don't have a happy ending slide for you today, but I do know that there are a bunch of dedicated laboratorians right here in your state working hard on this problem and many other public health problems, trying to keep everyone safe. So I know we covered a lot of information. I think I'm ready to open up for questions.
Jason, I have a question. Do you know of any new class of antibiotics being developed to fight these superbugs?
So yeah, antibiotic development is definitely one of the big problems in this whole story that I didn't really get to in this talk. So that the kind of the immediate success of antibiotics in the 50s, 60s, and 70s, people kind of stopped looking for new antibiotics in the 80s and 90s. So we've really tailed off on anybody kind of coming up with new drugs. Now that this problem has gotten so big and out of control, different companies are trying to come up with new drugs. A lot of the new drugs, like to treat carbapenemase-resistant bacteria, are really like different formulations of old antibiotics and an enzyme inhibitor to try and stop that enzyme. So not really new classes. And I don't think that there's really a lot on the horizon, but that's definitely another thing that needs to be worked on to try and solve this.
So if somebody thinks they have food poisoning, do they call their physician and say, I think I have food poisoning? Do they go to urgent care? What should one do if they think they have one of these villains?
Yeah, if you have food poisoning, usually, yeah, you would try and talk to your doctor and see, like I said, they may collect a sample. Some foodborne bacteria are just self-limiting. So sometimes it's just you stay at home and get better. And really a lot of foodborne diseases are viruses. So there's not really much you can do. You just have to ride it out.
Awesome. Looking to see if we have any other questions coming in. So this would be a point for our listeners out there. If you have any questions for Jason regarding superbugs, you can type them into the comment section on our live stream. We'd love to hear from you. And so with Mrsa, what was in the press a lot a couple years ago, it seemed like there were all these outbreaks like in schools with their wrestling programs, and yeah. You don't hear too much about it anymore. Have we taken care of that, or we just don't, it's not the new thing, so we just don't hear about it?
I think it's more that it's not the new thing. And kind of an interesting thing happened with Staph Aureus that so oxacillin and methicillin, the M in Mrsa, methicillin-resistant Staph Aureus. The next drug that's kind of the last line drug is called Vancomycin. And actually Michigan was the epicenter of a couple vancomycin-resistant Staph Aureus. And that was truly scary because that if we don't have vancomycin to treat Mrsa infections, then we're really out of drugs. For some reason, those strains have just kind of gone away. And that's a kind of an interesting thing about antibiotic-resistant bacteria is sometimes they carry these mechanisms that make them resistant to antibiotics, but that comes at a fitness cost to them, that they don't function as well expressing these genes. And that I haven't read what people think happened to the these vancomycin-resistant Staph Aureus, but that actually in that 2019 CDC antimicrobial threat, Mrsa was downgraded, and we're not as interested in that at a public health level because they just don't seem to be circulating. So it's one of those things that it just kind of disappeared.
Well, that's a good thing.
Yeah. But it could always come back, and we do still have a PCR test for that at the state lab, and we're ready to test for it if it comes back.
I know one of your things that you mentioned to decrease antibiotic resistance problems was to take your full dose of antibiotics when you're given them by your physician. Can you kind of explain why that is? I know we hear that a lot. It's on the bottle, make sure you take the entire prescription, but why do we need to do that?
Yeah, so bacteria exist in these large populations where they're going to kind of span the spectrum of kind of resistant to mostly susceptible. And if you only take a short course of antibiotics, you may not kill those or stop those bacteria that are a little bit more resistant. And the longer you let those more resistant bugs hang out, they're going to keep growing and repopulate. So you have to take your whole course of antibiotics and hopefully kill all of that bacteria that we were targeting in your infection, so that you don't leave any stray resistant bacteria hanging around.
Great. Catherine, do you have any questions for Jason?
Yeah, I do. Jason, I've read a lot about E. coli outbreaks that are linked to romaine lettuce or different types of lettuces. Why does this seem so common with lettuce?
So lettuce, a lot of times that E. coli comes from the contaminated water sources that they're using to irrigate their fields with. And then things like lettuce are kind of a perfect vector for some of these bacteria because they're grown on the ground where there may be dirt or feces, animal feces, and then you can wash it, but you don't cook it. So oftentimes that's why they want you to cook your ground beef to kill all of the bacteria in the beef that may have contaminated the food, but you don't often eat salad. It comes straight out of the field, straight onto your plate. So that's why lettuce oftentimes gets tied up in these E. coli outbreaks. And oftentimes they'll go out and they'll that's the theory, is that usually it's a cow pasture kind of upstream that may not be treating its water, its wastewater the way that it should, and that gets onto the field, and then what's in the field goes on your plate.
Mm-hmm. Is there a way to sort of track exactly where it's coming from, or? Yeah,
so state public health labs and the CDC, and we all try and track back if you do eat that contaminated product and we get your bacteria into our lab, we will look at its DNA and kind of get a DNA fingerprint of that bacteria and try and trace it back to others. And then you'll probably get a phone call from an infection control person and you'll say you ate salad. Other people with that bacteria will say they ate salad. Sometimes they'll go out to the store and get bags of salad to try and test those and see if we can grow the E. coli from the bagged salad. And then obviously if you get a positive from the food product, that would have, the supply chain information on it, and you can track back to hopefully find the field that this came from.
Could you talk a little bit about how you got here, what your background is, what you studied in school, and sort of what inspired you to take this path?
Yeah, so I went to Michigan State. I graduated from the BLD program in clinical laboratory sciences. I worked at a hospital lab for 5 years after that. And I thought, well, I saw this job open up at the state public health lab, and I thought public health microbiology sounded really interesting. So I've been at the public health lab now for 8 years. I've got to do a lot of interesting testing, a lot of different things there, had some really great experiences. But I guess I got interested in laboratory sciences, I guess in college when I was trying to pick a major or decide what I was going to do with my life. I knew I wanted to help people. I knew I liked science. I very briefly thought of maybe nursing, realized that may have not been for me, maybe try and find a more behind the scenes science profession. And that's how I kind of stumbled into clinical laboratory sciences, which I guess has been a perfect fit.
Very cool. Well, thanks so much for joining us today. It was really insightful. For everyone watching again, if you have any questions, please feel free to type those out in the comment section so we can answer them during the live stream. And Jason, if viewers want to learn more about this topic, where might they turn to dive in again?
I guess you guys have had a lot of questions about foodborne outbreaks. The CDC website, a lot of the different pathogens all have their own website. So I think it's cdc.gov/E. coli. E. coli has its own website that you can track foodborne outbreaks for E. coli. Listeria and Salmonella also have similar websites that all those are on. Like I said, that CDC antimicrobial resistant threat pamphlet, that would be an interesting resource.
Great.
Fantastic. I had a question, but I lost it out of my head. So maybe it'll come back around.
For everyone tuning in, again, this is our afternoon science snack with MSU Science Festival. We're doing this for a couple more weeks, so if you go to our Facebook page and click on the events section of our Facebook page, you'll be able to check out what we have in store for the next coming weeks. So yeah, thanks again, Jason.
Thank you.
All right, everyone, until next time.